So what our research is about is what we call cellular memory. It's the ability of cells, and not just brain cells, but cells in general, cells of our body, every kind of cell in the body, even single cells, microbes, organisms that only have one cell, the ability of a cell as a unit of biology to retain information, to register experiences, to have those experiences leave marks on the cell that last for a long time, and then those marks feed back on how the cell operates in the future. So, one way to put what memory is memory is from the past, but it is for the future. And that is true for not just our brains and our behavior as animals, where we go, what we eat, but for the behavior of individual cells. Each cell, even if it's a kidney cell or a gut cell or a bone cell, it has its own experience. It has molecules passing through it, liquids traveling through that cell, nutrients, waves of hormones, signals from other cells, and it's unpredictable and chaotic. And at every moment of time, it's barraged with hundreds of signals, and it has to make sense of them. It has to distill what is important about them and what should leave an imprint in that cell for the future. And that's what we call cellular memory. And what our research shows is that that cellular memory, this ability of any cell to register patterns of experience and leave within itself lasting imprints of those experiences, the way that a cell does that is the same way in which our brain cells register our experiences, what we call, normal memory, what we experience as memory, what happens in our brains when we respond to our experiences, when we learn something, when we form a memory. It is the same biological process that happens in each of our cells when they are confronted with their experiences, gut cell experiences or kidney cell experiences. So it's this continuity between different forms of memory, including brain memory, but not limited to that brain memory. That is the focus of our research. What we specifically looked at is this well-known property of memory. It's called the mass-spaced effect. So that's been known about memory in humans, in animals. Any system that you look at, any brain will have this property called the mass-spaced effect. If you have the same amount of training, learning, studying for an exam, you study for the same amount of time. If you do it all in one go, if you cram that learning into one session, you will form a worse memory than if you space it in time. Even though you study for the same amount of time, if you space it with intervals, your memory will be superior. So that's true for humans, for sea slugs, for mice, for any kind of memory that you look at. But that's never been seen outside the brain, in cells like kidney cells. And what we have shown by giving them these artificial stimuli, by stimulating their molecules in an artificial way, and then watching their memory genes get turned on by watching this reporter gene, what we've shown is that they also have this mass-spaced effect. The same amount of chemical stimulation delivered in one go produces less activation of the memory gene than same amount of training, just delivered in spaced intervals. So the difference is really just a few minutes. It's a few minutes of how you distribute that chemical signal, but the cells are able to tell the difference and respond to one protocol in one way and to another protocol in another way, just like our brains do to spaced versus mass stimulation. And we also show that the biochemical components of this response are the same as they are in neurons. So the same process is run by the same molecules in generic cells as what's happening during memory formation in the brain. So we think that that's evidence enough to say that it's all one process. It's not like memory. It's not similar to memory. It is that same process. Brains have it, kidney cells have it, all cells have it. It's much more fundamental than the distinction between brains and non-brains. I think that the correct answer to be here is to say everywhere and nowhere in particular. It is in the very process of conducting that signal, in the entire cascade of that that conducts this information, that the information is stored. It doesn't modify just one point or another point. It modifies everything. It modifies the entire state of a cell, and that it will include phosphorylation of proteins, turning some proteins on, some proteins off, turning on genes, those genes producing products that then go on and modify proteins again, that then go on and turn on other genes. It's loops and feedbacks, positive feedbacks and negative feedbacks, and collectively they modify the cell or an ensemble of cells for a long time, for a longer time than it took you to perturb them in this way. And for as long as that state is modified, we call that a memory. And when it comes back to its natural homeostatic state, we say that the memory is gone. And it might be that some part of that homeostasis has returned to the baseline, but some other part has not returned to the baseline. So it might seem that there is no memory. For example, if we're talking about a behavioral memory, about an animal finding, an escape from a maze, it might seem that the memory is gone, but you retrain the animal and the memory comes back faster and stronger. So it was there somewhere. Something was still modified. It just wasn't immediately obvious. So I think it's wrong to think of memory as something that has a particular place and that sits in there, and when it's there, the memory is present, and when it's gone, it's gone. We need to think of the entire brain as consisting of these moving parts, and every part can move in different directions for different durations of time. And when it moves somewhere, well, the brain thinks differently. And for the time that it thinks differently, we say that that's a memory. That is a great question. And I do think that there is a degree of preservation of information in these signals between non-neural cells. We see that in our work that, for example, the signaling molecules that are secreted from our kidney cells, well, after a particular stimulation, there'll be more of those molecules secreted from that cell, and so other cells in the vicinity would hear that molecule and maybe modify themselves in response as well. So it does exist, the modification of connections, communication between cells, and it's part of the cellular memory. But there is something that does distinguish neurons from non-neural cells. It's not that neurons are generic cells, they are special. And what makes them special is synaptic contacts. And it's not just the fact that those contacts can go into a far away location in the brain and send a message somewhere far away. No, that other cells can do that too. They can send chemical messages far away in the body. What makes neurons special is the specificity of those connections, is the fact that they can convey a signal not just to some cell in that part of the brain, but to one specific cell in that part of the brain, and then to another specific cell in a totally different part of the brain. And those two contacts can change independently of each other. That independent modification of those thousands of connections, specific connections, that is what makes neurons special, because it gives them a lot more degrees of freedom that they can modify. A generic cell can modify its gene expression. It can increase the expression of some genes, but not others. It can increase the production of some hormones, but not other hormones. But it can't increase a release of a hormone to that cell over there, but not to this cell over here. That it can't do. It can only modify it as a whole. But a neuron can do all of that and also modify specific connections with other cells. And that's what makes a brain a brain. It gives it more specific things to tweak, more dials to modify, more states in which to store that memory. So the capacity of the brain that's consisting of neurons to store information is vastly greater than the capacity of kidney cells or gut cells as we understand them today. Maybe we will discover in a couple of years that those cells also communicate through very specific, synaptic contacts, which is haven't discovered yet. But as of now, we don't know of that kind of degree of specificity in cells communicating with each other. That is still something that neurons and brains are very good at. But it's not to say that in principle it doesn't exist in other cells, and that collectively our body cannot store enough information for it to be meaningful for our lives.